Preparing a lab course is one thing, and providing a good lab space for the experiments for the students. Designing the course is a totally different story. And today I will let you into the details about how I designed this course, the yeast course, to have a very good learning experience for the students based on didactic principles. The two keywords are repetition and coherence. In this course, we build a stable and well-connected understanding of molecular methods that are relevant in any molecular biology lab, but also beyond the laboratory practice, gain an understanding of how we can translate yeast genetics experiments into medical application therapy screens. This lab course is designed in a way that it walks students through a series of experiments that are coherent. It's a type of mini-thesis lab experience where it's just a 2-week block of simple molecular biology experiments. We cover PCR, we cover genetic knock-in, we generate a new yeast strain, and we utilize the GAL system to make a sugar-inducible promoter, as well as we do genotyping by PCR and then analyze the transcriptional induction dependent on the sugar source by doing a Western blot for the tag proteins. I designed this course with modern science in mind. If a student came to a lab and started a thesis, a bachelor thesis for example, this would be exactly how they could imagine this works, just really squeezed into 2 weeks with a very simple workflow. So instead of just doing a PCR to look at XYZ and then go to a next experiment, which is somehow related but not directly, we really generate a yeast strain, analyze the yeast strain for functionality, and then do a real experiment with this yeast strain. And the central didactic element in this course is deliberate repetition. We learn best when we can repeat things, and we learn best when we have the time to repeat things. Repetition here doesn't mean doing the exact same more than once. It means variation of a theme. So for example, the course is designed so that there are PCR experiments twice. One PCR is a PCR to amplify a knock-in cassette, and the other PCR is a genotyping PCR. So it's not the same identical type of PCR, but it's a variation of PCR, giving the students the ability to think about what goes into a PCR reaction, what then are the differences between the two, when to use which polymerase, and then pipette them in the teams of 2 very independently the second time already. Everything else is also, if possible, included as a repetition. For example, the Western blot, we do just one Western blot, but the antibody development is done in 2 steps, so that they first look at an HA tag antibody development, and then they have the housekeeping gene the next day, so they can repeat the same steps and know then exactly what's coming, and they can independently repeat it the second time. So this repetition, at least 2 times with a little bit of variation, helps tremendously to learn. At the same time, the course is not just meant to get to know some basic molecular biology method. No, it's meant to be also incorporating what we do here into the bigger pictures, translate what we are doing in the yeast genetics lab course into the human system, medicine associated, and discuss in the lab report how this work in the yeast lab could be relevant for investigating human disease and coming up with an own experimental idea. Coherence is key here. The learning objectives, the experimental setups, course materials and assessments are all aligned. We have a 3-tier grading system. The students have the ability to experience here for the first time to write a lab book, lab notebook. And the aim is not to have the perfect lab notebook, but have it as best possible and learn how to decide what goes in, what doesn't need to go in. Is it complete? How to write such a thing anyhow, when they later on in the lab probably have to do it electronically. That will give a grade. The second grade is for a paper presentation. Each of them picks a paper, and then they will learn about the biological function of these genes of interest that we use here in the yeast lab course by reading this paper and presenting a small 10-minute presentation for their colleagues. They have a special guide, so they are not left alone how to prepare this presentation. The beauty is that each year the course is a bit different depending on the paper choices and the pick of the relevant key data that the students selected and presented to their audience and to their peers in the room. We Do not leave this isolated, the presentation gets a grade. However, we also gather together as a whole group and then we make a little whiteboard sketch of all the biological facts and features that they have extracted from preparing the paper presentation. So that benefits the whole group, that I'm not telling anything about the biological function of the genes of interest and the bigger context, but they give this information by presenting a scientific paper from the field. That is very rewarding because it's not just a presentation for another grade. It is really relevant to get the background information across of the biological context that we are working in. And the third level of grading is the lab report that the students write after this 2-week experimental week log by themselves. And this lab report is not only the report about the experiments with correct labeling, what's the result section, what's the discussion, but we also elevate the learned aspect to the next level by providing in the second week of the course more additional information on the concept of synthetic lethality, which is a relevant concept in medicine and in genetics to find targets and therapies to kill specifically cancer cells and leave somatic cells that are not cancerous intact. So to develop cancer therapies, the modern approach is to exploit synthetic lethalities, and what the students do in this course can be used screen for such synthetic lethalities. And to get an idea of this genetic concept in the yeast genetics context, but then also learn and discuss about how this could be used to really make a difference in the medical research field. That is very exciting, because we are a yeast genetics lab course here. However, everything is relevant for human health and medicine. And this is very pleasing for the students because they will then also write a paragraph in their discussion of the lab report about this concept and how they could use what they have prepared here in the lab course to find such new synthetic lethalities and figure out how to fight against cancer. So the repetition and providing the bigger picture of it all really gives a learning experience for the students that tremendously helps them to go into their bachelor thesis, which oftentimes comes directly or shortly after they have finished this module. Complementary to the other courses that the students are getting offered here, this is a great learning experience, and seeing a lot of different types of practical courses and didactic concepts is really valuable for learning also. This lab course is not just a set of experiments, but a carefully designed, coherent learning journey. Through the repetition of basic methods, with a slight variation each time, we have a deep understanding of the methodology and an understanding of the technique. If you're interested in designing a course for students that goes beyond doing one experiment and getting to know this experiment, or doing an experiment and getting to know a biological function, but connecting it all, this type of design might be something you would like to pick up and adapt to your own biological research. Feel free to leave questions, ideas and comments. I'm curious what you think about it. If you like this type of content, consider to subscribe for more teaching related content. See you in the next. Thanks for watching and happy teaching!